Hi, fam. Oh, oh, what was that?
Sorry, my purple jacket didn't survive the alcohol wash.
And honestly, they're terrible in social distancing. They can't resist hanging out in clusters together.
If you're really curious what's going on in the crazy world of prokaryotes, stay with me. I promise you, it won't be boring.
Wait, where is your nuclear envelope?
Nuclear envelope? I travel light! And no membrane bound organelles too!
So, all your inclusions live in the cytoplasm rent-free?
Unlike a true nucleus, the bacterial DNA lies freely in a region called nucleoid. The genetic material is basically naked, not enclosed by a nuclear membrane. And many bacteria also have smaller circular pieces of DNA outside the main chromosome. These are called plasmids. The plasmid DNA confers certain unique characters to such bacteria. Example, antibiotics resistance. You know how prokaryote play safe math? Well, they multiply by division. Bacteria come in various shapes. Spherical ones are coccus. In pairs, diplococci. In chains, streptococci. And in clusters like grapes, staphylococci.
And honestly, they're terrible in social distancing. They can't resist hanging out in clusters together.
Bacillus. We have rod-shaped bacteria. These are called bacilli. And this one seems to have arrived with a little sporting ambition. Some bacteria are beautifully spiral shaped, the spirillum. And apparently this one has decided that its flagella needs a little grooming. Priorities. And we have comma shaped vibrio because apparently straight lines were too boring. And these are just some of the fascinating forms bacteria can take. Okay, now that we have seen how bacterial cells look like, but what keeps these tiny cells protected? Let's start from outside and work our way inside. The outermost layer in many bacteria is called glycocalyx. If the glycocalyx is loose and slimy, we call it slime layer.
Basically, it's just loosely hanging around to see what sticks.
But when the glycocalyx is firmly attached to the cell surface, it's called the capsule.
So, in relationship terms, the capsule is fully committed.
Both slime layer and capsule are forms of glycocalyx. In 1884, bacteriologist Christian Gram developed a staining technique which is today known as Gram stain. He first stained them with crystal violet, then added iodine. Next step, he decolorized with alcohol or acetone, and last, he counterstained them with safranin. Bacteria that retained the crystal violet stain even after decolorization were gram positive, and those which lost the stain and took up safranin were gram negative.
Sorry, my purple jacket didn't survive the alcohol wash.
Inner to the glycocalyx lies the cell wall. It's made of peptidoglycan, also called murin. It gives protection and shape to the bacterial cell. And then there is Mycoplasma. It does not have a cell wall, so it does not have a definite shape.
Ooh, literally shapeshifter.
And inner to the cell wall lies the plasma membrane. It is similar to that of eukaryotic cell. It is selectively permeable and decides what enters and exits the cell. And wait, some bacteria have even more accessories, like long thread-like flagella, helping them to move. A bacterial flagellum has three parts: a basal body, a hook, and a long filament. Pili are elongated tubular structures made of special protein. Their role includes attachment, and some specialized pili are involved in conjugation tube formation for transfer of genetic material. Fimbriae are basically the clingy friends of bacterial world. Once they attach to a surface, they really don't want to let go.
Dude, I think I
Move on. You can't even let go.
Uh-oh.
Special membranous structure is the mesosome, which is formed by extension of plasma membrane into the cell. These extensions are in form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication and distribution to daughter cells. They also help in respiration, secretion process to increase the surface area of plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Now let's meet the protein factories of prokaryotic cell, the ribosomes. The prokaryotic ribosomes are 70S ribosomes made of two subunits, a large 50S subunit and a small 30S subunit. And before you say, wait, 50 plus 30 is 80, these ribosomes don't care about your math. That's because S stands for Svedberg unit, a measure based on how particles behave during centrifugation, not simple size that can be added. And when several ribosomes attach the same mRNA, they form a chain, a polysome or polyribosome, allowing that single mRNA to be translated into proteins. Inclusion bodies. Inclusion bodies are reserved materials stored freely in the cytoplasm. They are not surrounded by a membrane. They include phosphate granules, cyanophycin granules, glycogen granules, and gas vacuoles. Some photosynthetic bacteria use them in tiny flotation devices, helping them to move to a position where they can get better light. So that was bacterial cell. Tiny, simple, but not boring. And if you enjoyed exploring the bacterial world with us, tell us in the comment which part you like the best, and please don't forget to like, share and subscribe. Also, I have some MCQs for you. The answers are given in the comment section, in the pinned comment. Do attempt those, check the answers and tell us your score in the comment. Thanks for watching.